Yo, what's up, guys?
Welcome to our podcast first episode.
Omics to the rescue!
And I'm Clement, and joining me today are Ashby, Darren, and Jing Jie. So now we are going to explore how genomics and proteomics are going to improving the modern agriculture. So do you know that modern agriculture has helped us a lot, like providing better, more food, better health, and less poverty. So, but right now it's facing some big challenge.
Yeah, you're absolutely right, Clement. Now, there are many serious challenges ahead. The world population is rising very fast now, which means we nearly need almost double the amount of food that we produce today. On top of that, climate change, pests has made it even harder for crops to grow. Now, this puts extra effort in modern agriculture. However, good news is genomics and proteomics are powerful tools that can help us tackle these problems. That let us improve crop production and also make them more resilient to stress like drought, heat, and diseases.
Okay, let's take a look at genomics first, with the canonical examples like wheat. So the gene named RHD are actually responsible for reduced plant height. So this will significantly improve the lodging resistance. This means that the tendency of tall plants to fall over due to wind or rain. So the shorter stature actually enhances the photosynthetic efficiency by enabling a denser planting and reducing competitions for sunlight. And the shorter plants also are less prone to shading. This allows a more uniform ripening and also help with harvesting, which makes it more effective with uniform plant height. And the reduced biomass of the nutrient means that the nutrient will directly channel to the grain instead of the stalk growth.
I see.
So for another example like golden rice, it's actually an example of biofortification. It improves the nutrient content by incorporating the gene named phytoin synthase and also carotene desaturase, which is both genes are actually coming from maize and also soy bacteria. So it helps to provide up to 50% of the vitamin A uptake in single serving of the golden rice only.
Wow.
And there's these examples which is researched by Cavusoglu. It actually discovered this gene's named SINHX, synhex gene, which is actually improve the salt tolerance. So these genes actually plays a key role in maintaining the cellular ion homeostasis. So scientists, they actually engineer tomato plants with this kind of genes expression, and they have really successfully created cultivars that can maintain the fruit yield and quality even under a high salinity condition.
Oh, that's interesting.
That's very interesting, and I think the incorporation of genomics in plants is a very interesting and very revolutionary thing to consider. And it's kind of fun to see how these scientists are able to play with specific genes which are able to change or even enhance the crops. But I got one question which I think is still on my mind is how is it done? What's the method and how do they do it actually?
Hey, that's a great question, and I have exactly the answer for that. Now let's take a case study for example. There is this submerged tolerance rice research done by the researchers from University Kebangsaan Malaysia, UKM. The researchers uses a technique known as a marker-assisted selection, or more commonly known as MAS. So MAS allows breeders, the researchers, to pinpoint specific locations of traits, like for example maybe like stress tolerance, by using special DNA markers, which are usually SNP, single nucleotide polymorphism, or RFLP, restriction fragment length polymorphism. So the beauty of this method is that breeders can actually select the plant with the desired genes by just looking at their DNA. They do not have to wait for the plant to grow and show the phenotypic trait in the field. So the methods is rather, I would say, time saving and save a lot of money actually, because they don't have to grow the plant, right? So the maintenance are free. Now here's how it works in details though. So they started by focusing on the gene known as SUB1 genes, which are specific for and responsible for submerged tolerance of rice. So to find these genes inside specific plants, they will use DNA markers, like mentioned earlier, the marker-assisted selection. In this case, it was RM219 and RM23915. So these two markers are actually located next to the SUB1 genes, which means that when this marker shows up in the plant's DNA, it means that the plant also carries the SUB1 genes. Now with this information, they can later back cross these plants with the local plant varieties, which this method can bring the submerged resistance to the local strain as well.
Oh.
Now, this is a very elegant and highly efficient method, but most importantly, it gives farmer a practical and resilient rice variety that can survive for a long period of time under flooding conditions, making their harvest more secure even when the rains just doesn't let up.
Oh, that's
Wow.
fascinating.
Yes, that's
Interesting.
Yeah, right?
That's a great example of how genomics can be applied.
Yeah.
So actually, I also know there's one method that can be applied in genomics, which is
Oh.
known as genomic selection method. Yeah,
Okay.
that makes everything easier and smarter.
Mhm.
So instead
Smarter.
of instead of waiting for the plant to grow and checking its threads, so scientists can actually use this method to look at the plant's DNA, kind of like checking the blueprint before building something.
Oh.
Yeah. So and also predict how good the plant will be. And what's even better is they don't just look at one or two genes, they look at the entire plant genome. And now I want to show you how this method is very strong. Yeah. So there was a study in 2015 where they used this method to develop the drought tolerant maize.
Mhm.
And they used the entire plant's DNA to predict which one would survive the best in the most tough condition.
Oh.
Yeah.
I see.
So and it
Very
really
specific.
worked. So the maize they selected using this method gave about 7.3% more yield compared to the traditional one.
Wow.
So
Okay.
it's very interesting, right?
Yeah,
Yes.
the method that
It's
you mentioned is
definitely
very
interesting.
interesting.
Very interesting.
And actually, I've been thinking about what you said about the submerged tolerant rice
Oh.
that you mentioned earlier.
Oh,
Right?
yeah, yeah.
There's actually another incredible real-world success story, which this researcher named Xu, he actually combined the marker-assisted selection with the mapping and also pinpoint the sub-1A genes, which is basically a gene that serve as a master switch for flood tolerance in rice.
Oh.
And this has led to the development of new variety called Swana Sub-1.
Wow.
And what amazing is that this variety actually behaves almost identically to the regular Swana under normal condition, which will produce up to 5 to 6 tons of the rice per hectare. But when flooding hits, the original wild type Swana will have a survival rate of 0%, which means all of them will be
Die.
there. Yeah. But in contrast, this Swana Sub-1 can survive up to 95%.
Oh, that's a
Oh,
lot.
Half the
high.
time. Yeah. So that's a massive improvement just by incorporating one single powerful genes.
Yeah, very large difference.
Mm.
It's insane.
Oh, such details of data interpretation, Darren.
Yeah.
Actually, right, the same kind of data interpretation can also be applied to the drought tolerant maize we talked about earlier. So I remember there's one research that I read earlier. It was from 2015, if I'm not mistaken. The researchers uses something called genomic estimated breeding values to predict whether the plants have the best chance to survive under drought. So this method, right, instead of growing hundreds and thousands of plants and hoping that maybe one of them will have certain amount of drought resistance, they just scan the DNA and run the prediction. Now, the best part, the hybrid selected using this method yielded 7.3%, like it's like exactly the same as the one Clement mentioned just now,
Yes,
7.3% more
yeah.
than those of
That's
conventional
amazing.
mints.
Mm.
Now, time saved, yield boosted. Tell me what could have been better?
Yeah, it's
Yeah,
best.
you are right, Jing Jie. But don't forget,
Oh.
the data itself didn't act alone.
Really?
Yeah, really. Scientists had to interpret those genome-wide patterns,
Mm.
like not just only spotting a single gene.
Oh.
Yeah. So this kind of decision making really shows the strength of the genomic selection for the complex threat like drought resistant. So which don't rely on just only one marker.
Yeah, that's true.
Exactly, I think it also makes the process scalable. So with high throughput genotyping platforms, I think these breeders are able to run this kind of analysis on hundreds of samples at once. And I think with the right interpretation methods, right, they can prioritize the most strongest genetic profiles fast, which is very important and key when we're facing climate changes. And you can just imagine the ripple effect where farmers no longer need to gamble on weather and they can get varieties tested for harsh conditions. Plus, with reliable data interpretation, researchers can tweak these processes year by year based on results in the field, which ultimately it becomes a feedback loop that gets smarter every cycle.
You are right, Ashby. So we have been hearing a lot about genomics in agriculture.
Yep.
Yeah. Could you tell us what are the real economic and environmental benefits when we apply genomics to the crop improvement?
Yeah, I'm
That's
curious
a
about
great
that too.
question, I think. I
Mm.
think that's very interesting. So economically, genomics enables the farmers to cultivate higher yielding disease-resistant crops, leading to less crop loss and I think a more stable profit.
Yeah.
Environmentally, These
Okay.
improved crops, they typically require fewer pesticides and I think less water,
Oh, that's
and
very
thereby
important.
reducing the overall impact on ecosystems. Additionally, it also by selecting for traits such as drought tolerance or the ability to thrive in suboptimal soils, this genomics really helps the crop to adapt to climate change while enhancing the long-term sustainability of the food production.
Oh.
Oh, I see.
Alright, now since we have seen how genomics can pick the best gene in the crops, but so what actually happens in the plant?
Actually, right, so genomics, it shows us how like which genes are present in the plant, while proteomics can actually show us how the genes actually work. Now this is important because genomics is like an instructor, while proteomics is like the workers. It's something that is continuously working on. So it's like taking a look at the action plan of the plants actually. So when it's dealing with situation like drought or high salinity. So for example, right, so when plants are dehydrated, proteomics can then detect the rise in specific proteins such as maybe dehydrins, which helps it to survive during dry conditions. Now under salt stress on the other hand, you will find more transporters and antioxidant enzymes doing their job. So interesting, isn't it? Besides,
Very
proteomics does not just understand like stress tolerance, it is also vital for food quality and safety.
Oh.
In fact, the combination
Agreed.
of genomics, proteomics, and the yet to come transcriptomics have provided a 360 view of the crop performance and resilience. But I have a question actually. How though? How did they do it?
Yeah.
Okay, Jing Jie.
Right.
Actually, I know there is a method called 2D gel electrophoresis.
Okay.
Basically, it separates the protein based on their charge and size.
Mhm.
So it is pretty good at spotting big changes in protein, especially when the plants are under stress. Yeah. So, but on its own, it is not perfect because it can miss some even tiny proteins. Yeah.
Ah, okay.
So what scientists usually do is they pair it with the mass spectrometry and this combination cuts out the protein spots from the gel and then break them into smaller pieces and then use the mass spec to figure out what the protein exactly are. So I want to tell you guys that researchers had used this combination to identify the protein called OSCAM1 in the salt tolerant rice, which is the calcium sensor that activates a range of downstream gene. And this protein helps the rice plant to deal with the salty condition better. So anyone know about any other method?
I think I know one or two methods here. So,
Okay.
building on what we have learned, right?
Yeah.
I'd like to shift our attention to the quantitative side of things. So, modern proteomics basically leverages advanced labeling methods. Some examples, two of them I can give is ITRAQ or iTrack and TMT.
Ah,
So, for
TMT.
instance, there was this researcher named Ren. He used iTrack to analyze maize seedlings under drought stress and uncovering hundreds of proteins that react to dehydration. And I think these isobaric tagging methods are revolutionary because they allow us to multiplex several samples in one mass spectrometry run, which I think drastically improves the efficiency and also reduces the experimental variation. I'm kind of curious, so based on the topic we have today, how do you all feel with this advancement, how it complement the traditional methods that we have discussed before this?
Ah,
Well, that's a great point that you bring up here. Actually, the advancement of the current tools has actually covers the bad part of the traditional tools. But I find it particularly interesting is how these labeling methods, even though they are very powerful, but I feel like they are just one piece of the puzzle only. So there's also this method called label-free quantification, in short LFQ. Well, it relies solely on the spectral counts or the IO intensities over multiple runs. Even though LFQ might be slightly less precise than the technique like you mentioned before, like TMT, ITRAQ, but due to its simpler workflow and the cost effectiveness, it makes it an excellent tools or excellent method for large-scale studies.
I see.
So imagine running studies across hundreds of crop samples without the cost overhead of chemical labels. Like, wow, this is just amazing. But this flexibility is essential when it comes to scaling up experiments in crop proteomics, especially when you aim for a robust, comprehensive, and real-world applications.
yeah, what Darren said is more logical and realistic when compared to ideal situations. Now, the cost is something that we really just can't ignore in research, especially
agree.
in large-scale crop studies
Mm.
like mentioned by Darren earlier. Every expenses matters. So it's exciting to see how we have a suite of tools at our disposal, the ability to choose between isobaric tagging like mentioned by Ashby, which is accurate, and the one that mentioned by Darren,
LFQ.
which is label-free quantification, which is rather cheaper, means that these researchers, they can actually tailor their approach based on the study requirements and
Yes.
the budget constraints.
Yeah.
Cost does matters.
Mm.
Moreover, recent breakthroughs such as like I remember reading on single-cell proteomics and mass spectrometry imaging or MSI
Yeah.
are adding special dimensions to our understanding. So
New tools.
MSI, for instance, it allows us to pinpoint exactly where the tissue of certain proteins are up or down regulated during
Oh.
stress response. Well,
So
I
many
remember,
information.
Clement,
Yeah.
given your experience in classical techniques, right? How do you see these innovations changing the way we study plant stress?
Okay.
Mm.
Alright, JJ. Actually, traditional methods like 2D gels only gave us a big picture view by separating proteins based on their physical properties like charge and size.
Mhm.
So, but they lack the depth of the stress response at the very granular level. Yeah. So, but MSI builds on that by specially mapping protein distribution. It's like painting a detailed picture of how a plant reacts right down to the cellular neighborhood.
Good description.
Yeah. So
Mm.
this method is very important to develop the crops that are not just resilient, but also optimized for varying environmental conditions.
I think I absolutely agree with you, Clement. And I think this sort of system level view is kind of a game changer. So when you integrate these multiple analytical approaches, right, we move just beyond merely cataloging proteins to understand their functional networks during stress. And I think this kind of comprehensive insight can inform breeding strategies and also guide precise interventions to bolster crop resilience. And I think for each method that we have mentioned before, whether it's the precision of isobaric tagging, the cost effectiveness of LFQ, or the spatial clarity provided by MSI, it all plays a critical role in the overall framework for functional plant proteomics. That is one thing that's important as well. Darren, so I would like to ask you a question.
Yes.
What do you see as the benefits as we integrate these cutting edge methods into the context of, I think, food safety and quality?
Well, it's inevitable that proteomics actually brings a very big part when it comes to food safety and quality. You know, the stakes are high nowadays in ensuring food safety and quality, especially given our growing global food demands
Yeah, I see.
and the proliferation of intensive farming and also genetic modified crops, GMC or GMO. So one of the biggest benefit I would say is of integrating proteomics into food safety is precision detection. For example, in peanuts, we now know that the proteins like RR1 and
Mhm.
RR3
Mm.
are the major allergens to those who are allergic to peanuts. And these allergens, they stay active even after the roasting process. So
Oh,
using
okay.
proteomics
By using
techniques, we can actually detect these specific proteins at a very low concentration in the processed food. And it's not just only peanuts. We have to take wheat in for instance. There's these gliadin proteins, which are the key component of the gluten, and are responsible for triggering immune response in people with celiac disease. So with targeted proteomics like SRM and MRN, mass spectrometry, we can actually monitor the gluten-derived peptides with a pinpoint accuracy. So now we are not just guessing whether the food is gluten-free or not.
Mm.
We can actually
Precisely.
confirm 100% it down to the molecular level.
Wow.
Yeah, so this kind of sensitivity is a game changer for people with severe allergies. It means that food labels can be based on molecular evidence and not just ingredient lists, making the food system much safer.
I actually agree with that, and I think just beyond allergens, proteomics also help us with traceability and contamination too. So for instance, in cases of bacterial contamination, right, like E. coli, we can identify unique toxin-associated proteins using MALDI-TOF mass spectrometry. So what does this do is that basically it helps trace the contamination source. So whether it could come from the field itself, the packaging, or the transport. Plus, we're also discovering beneficial proteins. Some plant-derived protein peptides like those from soy or chicken pea proteins have these sort of antioxidant or anti-hypertensive effects. So at this point, I don't think we're just identifying risks, but also enhancing the functional quality of the food.
Exactly. What mentioned by Darren and Ashby earlier has big implications on how we will design and improve our crops in future. So, if we know that each proteins can enhance certain nutrients, for example, like increasing lysine contained in maize can reduce by reducing alpha-zein storage proteins, then we can target those traits directly in our breeding programs. Now, this is great. Similarly, if we want to identify like harmful proteins, for example, like those phytohematoagglutinin in kidney beans, we can just screen different varieties and optimize processing techniques to lower those toxin levels even before they reach the consumers. So, by integrating these cutting-edge proteomics tools, as mentioned earlier, we move towards food that's not just higher yield, but safer, healthier, and most importantly, sustainable. Now, this is where the future of agriculture is heading. It's heading towards this direction. Using data-driven insights, we can improve crops at every level, like Darren mentioned, the molecular level of all the plants, all the way to the denoplane.
Wow, that's great. Alright, thank you, Jing Jie. So in overall, Omics isn't just a research tool anymore. It's an integral part of the sustainable agriculture. So this is because genomics help us to figure out which plants genes are linked to the good traits, and proteomics looks at the proteins in the plants. So and helps us to understand how plants react to stress or pests, and then develop a more productive crops. So let's wrap up our episode of Omics to the Rescue. So we hope you guys found it informative and inspiring. And goodbye, and see you guys in our next episode.
Bye-bye.
Thank you.
Thank you. Bye-bye.